Well, thank you, everyone. And thank you for the invitation to come be part of this conference. It's my pleasure to be here. And I am happy to see so many of you in the audience considering the weather outside. So good for you for venturing out in this kind of weather. I'm here today to talk to you about my research program that explores the intersection between how we respond to our dietary inputs. And from that perspective, I'm going to be talking about how the diet influences the microbiota that are present inside of our intestinal tracts, and then how it affects the metabolism of the microbiota as well as the human. And I do so in an attempt to try to explore our goals of improving intestinal and systemic health. So I think we all have to recognize that worldwide, I think it's pretty easy to say most of us are suffering with a variety of chronic disease challenges. Risk, luckily, for some diseases are in fact declining somewhat, but then there are other diseases such as obesity and diabetes that are increasing rather dramatically in many places throughout the world. Since the world's population is increasing fairly rapidly, we have to face that as an issue. That means, though, that even though some disease risks are going down, the number of people suffering from those diseases are increasing as well. And so as a result of that, we have an expanding financial burden, not only to the individual, but also the country's healthcare system upon which they live and survive on. Now, to give you an idea, a context to understanding my motivation for being in this area, I want to show you some data from the United States. We have loads of people who are suffering with heart disease and diabetes, millions and millions of people that are suffering with those diseases. Now, cancer is not as a high incidence rate as cardiovascular disease, but if you look at the number of people that die every year from cancer diagnosis, it's almost the same as who die from those massive numbers of people that are suffering with cardiovascular disease, making cancer a very high causation of death in the United States. We also have a growing number of people that are suffering with inflammatory bowel diseases, and I'll explain the importance of this in just a moment. The other thing that the United States is definitely known for worldwide, and I wish this were not the case, is the number of people that are dealing with obesity. The majority of our population now are overweight or obese, and this is a huge challenge to us. And something that we're trying to figure out how to deal with from the healthcare system perspective. Now, with that in mind, so that you know where I come from and what my country looks like from this perspective on health, I want to take us back to my area of study, which is colon disease and specifics. The large intestinal disease, the biggest issue that we have is, again, the number of people that suffer from colon cancer. So this is the number of people that are newly diagnosed every year with colon cancer. And of those, we have 52,000 people that die every year from colon cancer, making it the second leading cause of death from cancer in the United States, second only to lung cancer. An interesting analysis came out back in 2009, where Harris and et al. Worked together to try to estimate the risk of a variety of diseases associated simply with the success of our healthcare system, which allows people to age even further. And one of the interesting observations that they had is that colon cancer rates were going to increase by 52% by the year 2030, simply because we had more older people in our population. So that's a rather dramatic increase over a very short period of time. Now, the unfortunate side effect of our obesity epidemic in the United States is the fact that obesity is directly tied to the incidence rates of colon cancer. So when you start looking at the problems associated with having simply more people who are of age to develop colon cancer, as well as the enhancement of risk caused by obesity, we are looking at probably wiping out the successes that we've had over the last several decades of overall achieving a decline in the general population of the number of people getting colon cancer. We have already started to note in people under the age of 50 a significant increase in the risk of developing colon cancer, to the point that if you were born in 1990, that you have twice the risk of getting colon cancer as someone who was born in 1950, and if you were rectal cancer is 4 times higher if you were born in 1990 compared to someone born in 1950. So that captures some of the problems associated with the obesity risk that we have in the United States. So the question I hope to kind of throw out to you as potential solutions today and some conceptual issues I want you to think about when you leave here is how do we reduce these problems? Well, the obvious thing is we need to develop better healthcare interventions, ways of identifying people who have the disease earlier on so that we can remove colon cancer so that they don't die from metastases of the disease. Obviously, that's a big part of the medical care system. We also have to do a much better job of understanding the underlying mechanisms that drive these disease processes and recognize that it's not the same for me as it may be for you, that we have slightly different diseases. So colon cancer isn't colon cancer for everybody. It's a slightly different disease. This will help us to develop targeted therapies for individuals so that we can do a better job of treating those people. But the thing I really want you to think about is something that was brought out in the Healthy People 2020 report by the Department of Health and Human Services in the United States. In that report, they stated that now our national goal should be to avoid preventable diseases from occurring in the first place. Worldwide estimates are that approximately 60% of the incidences of colon cancer are truly sporadic and are due to problems in diet and activity levels of individuals, so that we could eliminate 60% of those diseases if we have an appropriate diet. The unfortunate side effect is very little has been done from the perspective of primary prevention, and that's what this means, primary prevention. Not treating someone once they have the disease, but actually preventing them from getting it. And that is the motivation for my research, is to try to find ways to do this, and that means coming up with combinations of dietary and behavioral modification in individuals in order to achieve the true goal of primary prevention. So now that I've given you an understanding of why I'm doing my research, I want to talk to you about the colon so that we're all up to speed, so that you know, depending on, you may not know much about the colon itself, so I want to give you just a little bit of a primer, kind of like Susan did in her presentation. The colon luminal environment, so everything that's inside the colon, is primarily derived from those compounds, obviously, that do not get absorbed in the small intestine. The vast majority of the nutrients that get there fall into the complex carbohydrates category, either dietary fiber, resistant starches, things along that line. However, if you're eating whole foods, let's say that you had a carrot or a piece of broccoli or a whole grain bread, you're dealing with something that has still structural integrity of that plant. And so inside the cells of that plant that have not been broken up by you chewing and masticating it are a lot of other smaller compounds that are not absorbed in the small intestine. So in effect, you wind up targeting the colon directly with what could be beneficial compounds. Now, the metabolic profile of the luminal contents is going to depend on two things. One, the microbiota that reside there. And as you can tell, there's a lot of them living with us. Current estimates are that there are about the same number of cells in our body that's us that are them. So there's quite a few of them there. We know by looking around the world that the distributions and relative content of the microbiota vary in the individual, and they vary with the diet that individuals consume. So it's possible to modify the microflora that are residing there. In addition to that, it's also dictated, the metabolic profile is dictated by the substrates that we provide to the microbiota upon which they live. So their metabolism is highly dependent upon the substrates we provide them. And that is one way that we dictate who is living there, because not all bacteria can use all of the substrates that get there. So they're very nutrient specific. So this brings us to the work that we're pursuing, and that is looking at fiber sources that are rich in polyphenols, these other compounds that are contained in the plants. They're not nutrients, but they're things that seem to have very strong biological implications for us. And so we've used a variety of animal models as well as humans to explore the impact of these sources in our diet on colon as well as systemic health. And the animal models that we've used have been a chemical model of colon carcinogenesis, as well as a chemical model of inflammatory bowel disease. And we've also worked in one study with overweight and obese human subjects. Now, for the rest of the talk, I'm going to tell you some results from our studies using one particular dietary intervention that we've used on a variety of different platforms so that you can see how the body responds to that particular intervention in the role of colon cancer, inflammatory bowel disease, as well as in obese humans. But it's not the only intervention that we've worked with, and we have similar kinds of responses for all those various different types of interventions. Okay, for those of you that are not familiar with colon cancer, this is probably the earliest visible lesion of colon cancer. They're called aberrant crypts. So what you're looking at is a colon that we've opened up, laid out flat, stained so that we can see the DNA in the epithelial cells. So that's all these dark blue circles that you see here. The little white spots are the opening of the physiological units called crypts in the colon. Now, obviously, these little guys, these are all the normal crypts that reside in your colon. And you and I, we both look the same way. It's not just our rats. And then you have these large features. This is a crypt. Just like this is a crypt, this is a single crypt. They're very enlarged. And we believe that foci of these aberrant crypts, like this large one here that contains about 16 aberrant crypts, are where colon tumors actually originate. So we can go through and visually quantitate the disease process in our animals. And so those are the data that I'm going to be showing you here on the next slide. So for the intervention that I'm going to talk to you about, we have used what referred to by a lot of people as an ancient grain, sorghum. It is consumed very widely in Africa, India, parts of China. It's now becoming found in a lot of the food products in the United States as well. But it has some interesting components. Some of the older varieties, like the black sorghum and the brown sorghum, are very high in polyphenol content. And most of the work that had originally been done with these was to characterize their antioxidant activity. And they have antioxidant activities that are way beyond what you find with the classic example of a blueberry as being a high antioxidant food. They're 10 to 20-fold higher in their antioxidant activities. And so we wanted to explore their impact on colon cancer to find out if there might be some benefit there. And what we found is that if the rats had been consuming a diet containing black sorghum or brown sorghum bran, so that we had 6% dietary fiber in the diet, we were able to significantly reduce up to 50% the numbers of these aberrant crypts in these animals. So that's a significant impact because in this particular chemical model of colon cancer, every animal will eventually get a colon tumor. And so we were able to reduce this at this finite point in time by 50% if they'd been consuming these diets. Now, we wanted to find out what the relationship was between the antioxidant activity in the diet, here indicated by the ABTS activity on the x-axis, and the number of aberrant crypts in the animals. And I was stunned whenever we plotted this, did the stats on it, and found that there's a really strong relationship between the antioxidant activity of the diet and the numbers of aberrant crypts. I don't want you to think that I'm saying it's the antioxidant activity that is causing the protection against this disease. But what this does tell us is that it's the compounds in the diet generating the antioxidant activity that is responsible for the disease protection. And those are the polyphenols that are unique to these various different brands. Now, in this experiment, we did what might be considered a poor man's way of determining if the microbiota had changed, and that's to measure the level of total short-chain fatty acids indicated in these beige bars, as well as the concentration of butyrate in the lavender bars. And what we found is if the animals were consuming a white sorghum bran, that there was a significant increase in both total and butyrate concentrations in the feces. However, if the animals had been consuming the black sorghum bran or the brown sorghum bran, there was a significant reduction compared to the animals that were consuming our cellulose-based controlled diet. This was a bit stunning to us because we've always associated an increase in butyrate concentrations as being considered to be a protective element against colon cancer. I will tell you that we have some measurements in these animals indicating that at least some of the active transporters for short-chain fatty acids, their expression was elevated. So it's possible that we're achieving greater transport or absorption of these short-chain fatty acids, and that may be how they're operating. But that's the subject of another experiment that hopefully, if I can get funding for that, for the proposal I submitted, we'll explore that as well. Now, one of the other things that we're very interested in are some of the processes that you see occurring in colon tumors. In colon tumors, you have very high levels of the pro-inflammatory compound COX-2 in the tumor compared to the normal tissue, and you also have very high levels of BCL2, which is an anti-apoptotic compound. And apoptosis has been found to be highly related to the development of tumors, much more so even than proliferation is. And so anything that we can do to enhance apoptosis, the better off we are with respect to eliminating tumors. And what we found is, regardless of which one of the sorghum bran diets the animals had been consuming, we wound up having a significant reduction in the level of both of these proteins. Okay? So that was very positive from our perspective. So again, if we're reducing COX-2, that means we're reducing the potential for inflammation in the colon, and by reducing BCL2, we should have an improvement in the ability of those cells to respond to apoptotic signals. And in this experiment, we did find that there was a significant increase in apoptosis associated, especially, again, with the brown and the black sorghum bran diets. Well, since we have observations indicating that the sorghum brans may be operating through an inflammatory type of pathway, we wanted to try to look and see what this source of fiber might be doing in an inflammatory bowel disease model. You know, were we truly modifying the microbial populations and their metabolism, and were we mediating inflammatory regulators to the point that we're able to suppress inflammatory bowel disease? And this is important because we know that a dysbiosis or a perturbation in the microbial populations is thought to be directly tied to the development as well as the severity of inflammatory bowel diseases. And importantly, people who suffer from recurrent bouts of inflammatory bowel disease are at a much greater risk of developing colon cancer. Some estimates are as high as 8 times higher risk in those individuals. So if there's something that we can do to reduce inflammatory bowel disease, we not only help individuals with that disease, but we also have the potential of reducing colon cancer as well. So here's some data from, again, another rat study where we provided animals with diets that contained either the black sorghum bran, the brown or sumac sorghum bran that I've shown you before, as well as another bran from a sorghum, a high tannin black bran. And we also included 2 control diets this time. We have a cellulose diet here, as well as a diet containing the fermentable fiber pectin. And the data that I'm showing you are for NF-kappa B activation. NF-kappa B, for those of you that aren't familiar, is a transcription factor that is very much so involved in the regulation of inflammatory gene expression. And then I'm showing you injury score, so actually characterizing the injury to the colon walls themselves. Now, the data that we have here are the change or the difference between the control animals and the animals that were giving the chemical in order to induce inflammatory bowel disease. So these values are differences. And what we see is that if the animals have been consuming the black or the sumac bran, as well as our control diet here, cellulose, that we have a very small change in the activation state of NF-kappa B and a correspondingly relatively small change in the injury score in these animals, especially in contrast to one of our control diets, pectin, which produced a considerable increase in both of those factors. Now, in this experiment, we did have a chance to sequence the microbiota to find out, at least in broad characterizations, who's living there. This color map I like using. It shows you a value. Each one of these columns is an individual animal. And you see that within a diet, there is some differences among the animals in that particular group. However, it's very easy for anyone to see that, in general, there are major shifts in the various different phylum in the feces of the animals that have been consuming these diets. So we've had a major change in the microbiota. Now, as we explore the depth of this, this graph shows us the distribution of Bacteroidetes. These are the hashed lines at the bottom of these columns. the firmicutes, those light gray areas above that, then some other category and unidentified microbiota based on our screen. And what we find is that the animals that were consuming any of the sorghum brands, whether it be black, sumac, or the high tannin black, that there was a significant reduction in the ratio of firmicutes to Bacteroidetes compared to the control diets. Now, the importance of this observation is that in individuals that have inflammatory bowel disease or are obese, that there is an elevated ratio of firmicutes to Bacteroidetes. So by all of our interventions, we're able to reduce this particular ratio to the point that it would be suggestive of an improvement in the state of inflammation and potentially with obesity. So this brings me to the next topic, and that is the issue of obesity. Obesity, we know, promotes diabetes, cardiovascular disease, and is highly tied to colon cancer risk. And as I showed you earlier, we have a huge problem with this in the United States. So it's something that we really have to understand better. We know that there are perturbations in microbial populations that are caused by diet. And there's now some argument that some of these microbial population changes may actually drive the risk of obesity in some individuals. So that led us to ask the question, can our bioactive compounds impact the microbiota in the colon of our overweight subjects in a way that beneficially alters the host metabolism? So we conducted a study using overweight subjects in a crossover design experiment. We have observations collected at baseline before they consumed our experimental treatment. The treatment was a breakfast cereal that we made from whole grain flour derived from the sumac sorghum, to which we had added additional bran so that we had an elevated fiber intake as well as the bioactive compounds that are found in the bran fraction. The individuals consumed either 50 grams of the cereal for breakfast or 100 grams. That was split 50 grams for breakfast and 50 grams later in the day. They consumed these for 4 weeks, and then there was a 3-week washout period, and then they consumed the other level after that point in time. The observations from this experiment included the microbiota in the intestine that we did by sequencing as well, and then we performed untargeted metabolomics using plasma samples from these individuals. So I'm just going to show you one piece of data for the microbiota. It's not as dramatic as our animal experiments because these people just had to change cereal. They didn't change their diet completely, so you don't see these massive shifts. But you see some shifts in particular microbiota that are, we think, very telling with respect to its opportunity to improve the health of these individuals. One of them being Faecalibacterium, which we found were increased if the individuals had consumed either the low dose or the higher dose of the sorghum brand cereal. So these scatter plots to orient you for these data as well as the ones on the next slide. The scatter here in the first 2 panels, those are the baseline observations. And then afterwards you have the lower dose and then the higher dose. And what you see is, this is Faecalibacterium here, that there's a significant increase with either dose of the sorghum brand cereal. Further, if you look at Faecalibacterium prausnitzii, which is the right panel of data, you see that there's a significant increase in the level of the detectable amounts of this particular bacterial group in the individuals who had been consuming the higher level of sorghum brand cereal. Now, the important part about this is that if you study individuals who have inflammatory bowel disease or are obese, you really have a hard time detecting this particular bacterial group. It doesn't show up in a high enough relative proportion to be a detectable level. With our dietary intervention, we had individuals that wound up having 15% of the relative abundance of the known bacterial groups being this particular bacterium. And so we were very successful at increasing this. But one thing I also want to draw your attention to is the base of these scatter plots. If you look at the beginning, before they start the treatments, we have a large percentage of individuals, again, that you cannot detect this bacterial group, either as a major group or as the smaller group here. You can't detect it. However, after they have been consuming the diet for a while, there are only a few individuals that retain an inability to detect this particular bacterium. Most individuals are starting to show that that bacterial group does reside there. It just had been suppressed by their diet prior to that point in time. So these are the untargeted plasma metabolomics data that I want to share with you. Again, just a few selections. In panels A, B, and C here on the left, these are all compounds that are known to be metabolites produced by the microbiota whenever they're metabolizing the polyphenolic contents of the diet. Okay, it's not something that you and I as mammals can produce. It's something that they produce. And what we see is that in this particular case, all of these compounds are significantly elevated after the individuals have been consuming either the 50 or 100 gram dose of our sorghum bran cereal. Now in panels D, E, and F are gamma-glutamyl amino acids, okay? In this particular group of molecules, we see that there is a significant reduction in the level of these particular compounds after the individuals have been consuming our diet. The importance of these observations are that low levels of compounds like 3-hydroxyhippurate that we have here, as well as elevated levels of gamma-glutamyl transferase activity that are responsible for producing these amino acids, are all associated with an elevation in cardiovascular risk and impaired glucose tolerance. This suggests that our simple replacement of whatever their existing morning breakfast cereal had been with our breakfast cereal had done something metabolically in a systemic way that would potentially contribute to a reduction in cardiovascular risk and an improved glucose tolerance in our overweight and obese subjects. Now, I've talked quite a bit about microbial metabolism, and one of the byproducts of microbial metabolism that has been studied for, let's face it, decades, and that's the short-chain fatty acids. We have shown data that I've shared with you here today that those polyphenol-rich fibers from sorghum are in fact impacting the microbiota and the short-chain fatty acid levels. Butyrate has been of special interest over the decades because it is the preferred metabolic substrate for the colon epithelial cells. So they prefer to use butyrate instead of glucose for their metabolism. In addition to that, and importantly, from the perspective of how it may be influencing colon cancer, is that butyrate is an epigenetic regulator of gene expression. And historically, we've known that butyrate is an inhibitor of histone deacetylases. And luckily, we had a couple of people who explained all of that for me yesterday, so I was able to remove that from my talk. So you guys know what that means. It opens up the chromatin structure and allows the promotion of expression of certain genes. And we've demonstrated that as well as others. Here I'm showing you some data from an experiment we conducted using the HCT116 colon cancer cell line. And we found that both the level of acetylation for histone H3 and H4 were elevated, with the elevation becoming significant for the H3 histone if the cells had been incubated with butyrate or incubated with butyrate along with the N3 polyunsaturated fatty acid DHA. So this kind of confirmed that in our hands, we're able to produce data that are similar to everybody else's data out there, that there is a change in histone acetylation. But we wanted to explore some of those other mechanisms of epigenetic regulation, such as promoter methylation that you heard about yesterday. And so our first measurement was to look simply at global DNA methylation status. And we found that, to our surprise actually, that butyrate was almost as effective at reducing global DNA methylation as our positive control, 5-AZA-DC. We did not expect this kind of response. We expected to see something, but nothing to this scale. And so this is the first observation in the literature saying that butyrate can do this. Whenever we combine butyrate with the fatty acid DHA again, there was some reduction, but at this point, there was not significant. So it was a very mild reduction in global DNA methylation. Now, our purpose in conducting this experiment was just to look at overall levels. We were actually targeting the methylation of genes involved in the regulation of apoptosis. And I will tell you there are a suite of about 15 genes that we looked at, of which there were about 7 that showed some significant changes in methylation state associated with our interventions. But the real question at the end of all this is, what happened to gene expression, right? Did we modify that? And so those are the data that I'm going to show you next. And here I've selected 2 of the regulators of apoptosis, TNF, I always forget it, RSF25, which is also known as death receptor 3, as well as DAPK1. And here we find that there is a 5-fold change in the level of expression with butyrate exposure compared to our control incubation for both of these genes. If we incubate with butyrate plus DHA, the levels are 3.5 to almost 3 times that of the control. Now, these are interesting because it tends to suggest, based on what we observed with the global DNA methylation data, that butyrate seems to be driving the expression of these genes much more so due to its impact on methylation than its impact on acetylation state, which really was surprising for us. And so we're trying to explore this a little bit more so that we can figure out what the underlying regulatory element might be that butyrate is impacting so that it can achieve this kind of expression. So this brings me to my conclusions. Polyphenol-rich fibers, as those that I've shown you today, do in fact impact the microbiota. And more so, they impact the metabolic products of the microbiota, which have an impact on colon health. I've shown you data demonstrating that they can suppress inflammation and that they can suppress colon carcinogenesis. In addition, I've shown you data that the microbiota that are present in the intestinal tract not only produce what we consider to be the standard byproducts of short-chain fatty acids, they also produce a variety of metabolites of these polyphenolic compounds that are now of a structure and a size that it's possible for them to be absorbed and have an impact on systemic health. Some of the outcomes of those modifications of those products are that they contribute to regulation of colonocyte transcriptional factors, and they have the ability now, because they are getting into circulation, to influence systemic physiology, where we demonstrated it may be possible to reduce cardiovascular risk and improve glucose tolerance even in overweight and obese subjects with this very simple modification to their diet. So what does it mean, though? Why is these observations important? Well, if I bring you back to the statement that we want to avoid preventable diseases from occurring in the first place, that's what's motivating me in my work, preventing it. That means we wind up hopefully reducing the suffering in the individuals that have these diseases, but also reduce the financial burden for the individuals and on the countries they live in. To do that, we cannot simply rely on therapies. Yes, we have to pursue those mechanisms, we have to develop effective therapies, but if we really want to do this, we have to also pay some attention to the other side of the equation. So, if our goal is in fact primary prevention, we have to determine which foods can confer these health benefits. We've been making recommendations to people for a long time now. Eat more fruits, vegetables, whole grains, eat a varied diet, but we don't tell them what to eat. If they're choosing to eat French fries as their vegetable, they're not really gaining a benefit from eating more vegetables, right? We have to tell them what they need to eat. And so for that, we have to do this kind of research. So I want to leave you with this thought. This requires the same kind of concerted effort and funding that we've put forth for drug development and medical treatments. Just like the therapeutic research deserves research effort, we have to put research effort and funding into primary prevention so that we can actually inform people what they should be doing in specific terms, not just broad generalizations. So with that, I want to acknowledge all the work of the great people that have been in my laboratory over the last few years that contributed to these observations, as well as my wonderful collaborators at A&M, as well as around the United States and Canada, and of course the funding agencies that provided the money, which otherwise, if they hadn't, we would not have been able to do the work. So with that, thank you very much, and I'll be happy to entertain any questions.